Hello members, you're much welcome in this session where we want to look at the cell envelope between gram-positive, gram-negative, and acid-fast bacilli. So I want to see how their cell envelope differ from each other. And as we start, we urge you to subscribe, like the video, and share to many others. So we are looking at different types, how we can type different bacteria according to their structure of cell envelope. So we want to see how the gram-positive envelope differ from gram-negative, and how does it differ from acid-fast bacilli like Mycobacterium tuberculosis. So the components of gram-positive, because we want to start by differentiating between gram-positive and gram-negative. And the major difference is in their peptidoglycan layer, because their cell wall, both gram-positive, gram-negative, and acid-fast, they are made up of peptidoglycan layer. So this peptidoglycan layer we are talking about, peptidoglycan layer, it is, if I split into two, it is made up of two words. It has peptide and glycan, whereby peptide means a chain of amino acids, maybe two or more. So it can be either a peptide chain or a polypeptide chain. Then the glycan, this comes from polysaccharides, which contains two types of the glycan, whereby we are going to see NAM and NAG, which is N-acetyl uramic acid and N-acetyl glucosamine. Those are the glucose molecules found within the peptidoglycan layer. So this chain is a chain of peptidoglycan layer, and it is joined together by the beta-1,4 glycosidic bond. So the beta-1,4 glycosidic bond joins one glycan to another. And we are saying the first one is always NAM, this is NAG, NAM, NAG, NAM, and NAG. So they alternate. You find you begin with N-acetyluramic acid joined together to N-acetyl glucosamine, and the bond which joins them, we are calling it the beta-1,4. So this bond which is joining them, we are calling it beta-1,4 glycosidic bond. This is the bond that joins these glucose molecules, which are part of the glycosamine glycans or the polysaccharides. Then it is, so we see N-uramic acid, N-acetyluramic acid, and N-acetyl glucosamine, they alternate in the chain. So if we have one chain and the second one, we see they join together via peptide bonds, and these bonds contain 4 amino acids. So this one we are going to call it tetrapeptide bond. Tetrapeptide, because it is made up of 4 amino acids, whereby it is made up of DR9, then glucosamine, DR9, glutamine, DR9. So this is DR9, glucosa, this is glutamine, this is lysine, and then this is DR9. So it starts with DR9 and ends with DR9. So it has 4 amino acids joined. That's why we are calling it a tetrapeptide bridge. And it is always attached to the side chain of N-uramic acid, N-acetyl uramic acid. So these ones, they alternate. So if we find another NAM, only NAM has the tetrapeptide bond. So NAG does not have a side chain of tetrapeptide bond or side chain. So these chains, which are made up of these different 4 amino acids, that is dealanine, glutamine, lysine, dealanine, these 2 side chains on NAM, they can be linked together. They are linked together via the lysine by a pentaglycine bridge. So it is joined together by pentaglycine bridge. We are calling it penta because it is made up of 5 glycine molecules, which we are calling a pentaglycine bridge. So we are going to find out that even this one is at the lysine, it joined together with alanine by the pentaglycine bridge. Even this one at the lysine position, it joins by the pentaglycine bridge. So this bridge that joins 2 side chains is what we are calling the pentaglycine bridge. And it is this bridge that enables cross-linkage of the peptidoglycan layer within the cell wall. So this is specifically the structure of cell wall, both in gram-positive and gram-negative and AFBs. But the difference is in the size, whereby we are going to tell you within the gram-negative, within the gram-positive bacteria, the peptidoglycan layer, which we are seeing here, is thick. This one has a thick peptidoglycan layer measuring between 8 to 20 nanometers. So this one is very thick with multiple layers of peptidoglycan, whereby this is the first layer, the second layer, the third, fourth, fifth, and sixth, and even more. So with the gram-positive bacteria, they have a thick peptidoglycan layer. That means it has multiple peptidoglycan layers. And this one describes the components of the peptidoglycan layer, made up of these peptidoglycan chains joined together by the side tetrapeptide chain and the side tetrapeptide chains on the N-muramic acid are joined together by the pentapeptide bridge, or sometimes you can call it penicillin binding proteins, or you can call it a transpeptidase, which joins this. And we are going to see drugs like beta-lactams, that is like penicillin, cephalosporins, carbapenems. They target by blocking the cross-linkage, whereby they target the pentaglycine bridge, preventing the cross-linkage of the peptidoglycan layer, and hence you cannot form a new cell, which is integral. So this is what we can talk about. So we have seen it has the first layer. This one is called the capsule. All of them have the capsule. So they have capsule or slime layer. Then we are seeing that the peptidoglycan layer in gram-positive is thick, meaning it has multiple layers. Multiple layers means greater than 3. That is what makes it thick. Even gram-negative, still they have what we call the capsule. Though gram-negative bacteria they are more capsulated than gram-negative. So still on this side we see they have capsule or slime layer. Some have capsule, others have slime layer, but which all serve the same purpose as we saw under the structure. Then still this one also has peptidoglycan. This one also have peptidoglycan layer. But where is the difference? This layer is thin. This one has 1 to 2 layers of peptidoglycan, whereas this one has greater than 3 layers. whereby they are between like 3 to 8 or even more. So that is the difference. So as we are seeing here, this one has 1, 2, 3, 6 layers. This one has only a single layer of peptidoglycan. So here the peptidoglycan in the gram-negative is just it has single layer, whereas this one has multiple layer. That is the major difference. Another thing we are going to talk about in this session is that in gram-positive bacteria, apart from the capsule, apart from the cell wall having thick peptidoglycan layer, this one is the one that makes the cell wall. It also has what we call teichoic acid. And this teichoic acid, which we can abbreviate as TA, it is these lines which are linking multiple layers. So we have this one, which we are going to call it lipoteichoic acid. Lipoteichoic acid. Then lipoteichoic acid, it is connected to the inner membrane, because apart from the capsule, the peptidoglycan layer which forms the cell wall, it has also what we call the inner cell membrane. And this cell membrane is made up of phospholipid bilayer. So the lipoteichoic acid joins from the glycolipids of the inner cell wall, inner cell membrane. That is the lipoteichoic acid. Whereas the ones that join from the glycan layer, we call them wall teichoic acid. So meaning the teichoic acid is divided into two. We have what we call lipoteichoic acid. Then we have what we call wall teichoic acid. We have seen lipoteichoic acid joins at the inner cell membrane, whereas wall teichoic joins at the peptidoglycan. But you can ask yourself, what is the importance of this teichoic acid, which is made up of sugars and some proteins? Its function majorly is it enables entry of cations, it regulates cation movement in the cell. But apart from regulating cation movement, it is also antigenic, meaning we can use it to type some gram-positive bacteria. It also prevents cell lysis. It prevents cell lysis. That is the function of teichoic acid, which is only found in the gram-positive bacteria. Whereas gram-negative, apart from its cell wall, which is made up of a single peptidoglycan layer, it has the capsule, because all of them have the capsule. It has more additional layer, and this layer is known as the outer, which we are seeing from here. So from here to here, we have what we call the outer layer, or outer membrane. There is outer membrane, then it has the peptidoglycan layer, and the space between the outer membrane and the inner, this is the inner membrane of the cell membrane. So the space is what we call the periplasmic space. It has periplasmic space. So we are seeing, on addition, this one has the outer membrane, which gram-positive do not have. And this outer membrane in gram-negative, it is made up of lipopolysaccharide. Whereby this lipopolysaccharide, another name you can call it an endotoxin. and its major role is protection. It acts as a protective barrier to the gram-negative. And apart from possession of lipopolysaccharide, it has also pollins. And these pollins enable entry of materials within the cell. So it acts as a protecting plate. So you can ask yourself, where are the pollins? We are seeing the pollins here. This is a pollen, meaning it has a pore. That is the outer layer. It is made up of, this is the pollen, this is the pollen, this is the pollen. Then it is made up of lipopolysaccharide, which are these lines indicated in red, whereby the lipopolysaccharide is made up of lipid A, then it has the core oligosaccharide, it has O antigen. And this O antigen also, we normally use it to type different gram-negative bacteria. Then the core oligosaccharide, so if I go to this lipopolysaccharide, where it starts is the lipid A, then it has the core, and its end is the O antigen, or what you can call O polysaccharide. So those are the three components of this protective layer, which we are calling endotoxin, or we are calling it a lipopolysaccharide, only found in gram-negative bacteria, whereas gram-positive do not have this outer membrane, which is made up of lipopolysaccharide and pollins that regulate entry and protects this cell. But you can ask yourself, what anchors this outer membrane? In this outer membrane, we are seeing, made up of lipopolysaccharides and pores, there is a lipoprotein layer. So these green substances, which are anchoring the outer membrane with the peptidoglycan layer, they are known as lipoproteins. So these are the lipoproteins. And the function of lipoproteins, so we see even containing lipoproteins. And these lipoproteins stabilize, provide stability to the outer membrane, and also anchors the outer membrane to the peptidoglycan layer. That is what we can talk about, the lipoproteins which are found in the outer membrane and enabling anchoring or stability of the outer membrane within the gram-negative. Then there is what we call the periplasmic space. What is that space? It is the space between this outer membrane and the inner membrane. And this periplasmic space, we have seen, it also contains scissor-like substances. And these scissor-like substances, we are seeing in the periplasmic space, they are known as hydrolytic enzymes. Hydrolytic enzymes like beta-lactamases, like penicillin-binding proteins, which plays a big role in catalyzing different reactions within the cell. So hydrolytic enzyme, one of them are like the lactamases, which are also found in periplasmic space, at the same time having our peptidoglycan layer and lipopolysaccharides, what we are calling lipoproteins. So that is how we are able to distinguish between. But what I want you to appreciate is that both have inner membrane, inner cell membrane. Even also this one has inner cell membrane made up of a phospholipid bilayer, having hydrophilic head and hydrophobic tails. And the function of the cell membrane is to regulate movements of materials within the cell of the bacteria. Hope you guys you're enjoying this video. I'm not confusing you so much, but in summary, if we ask you, state the differences between gram-positive and gram-negative. One, in a gram-positive, they have a thick peptidoglycan layer made up of multiple layers. That is one. But within the gram-negative, it has a thin peptidoglycan layer, the cell wall. And this thin peptidoglycan layer is made up of 1 to 2 layers, making it thin. That is the major difference. And that's how these ones are able to retain the primary stain, that is crystal violet, but these ones do not retain the primary stain during gram stain. Another second difference is that gram-positive contains lipoteichoic acid, whereas gram-negative lack the teichoic acid, which does different roles of regulating ion movement and being an antigenic. Then we see gram-positive lacking the outer membrane, which is made up of the lipopolysaccharide, that is the endotoxin, which acts as a protective barrier, together with the pollins, but gram-positive lack the outer membrane. We see the gram-negative having the periplasmic space, having hydrolytic enzymes like the lactamases, which the gram-positive lack. That is the major differences between gram-positive and gram-negative. And this gram-positive, it occupies between 50 to 80% of the dry weight. The peptidoglycan layer occupies that, but the peptidoglycan layer in gram-positive occupies 5 to 10% of the dry weight of the cell envelope. That is, so this percentage is for the peptidoglycan layer of the gram-negative. So that's why we normally say that the gram-negative bacteria, they are very porous. Imagine the outer membrane is made up of lipopolysaccharide, but at the same time it has pollins. So when I stain with crystal violet and I apply a mordant such as Lugol's iodine, it is not able to cross-link or to bind the crystal violet to this cell. So when I wash with alcohol or decolorizer, like acetone, I easily remove the crystal violet and it ends up taking safranin or neutral red. That's why they appear pink, whereas these ones appear purple on staining. So we are saying on gram staining, these guys, they appear purple on the microscope, whereas these ones appear pink. Why? These ones, they take safranin or neutral red or dilute carbofuchsin, whereas these ones retain crystal violet, hence appearing purple on a microscope after gram staining. Then, what of acid-fast bacilli? The difference between these two and the acid-fast bacilli, one thing I want to tell you that acid-fast bacilli are weakly gram-positive. These ones are weakly gram-positive bacteria, meaning their structure is the same. They also have a thick peptidoglycan layer. But the challenge is that for them, they have the outer membrane has what we call waxy mycolic acid. And this waxy mycolic acid prevents entry of the crystal violet during staining. So it is best demonstrated using Zn stain, whereby this Zn staining, we apply heat to dissolve the waxy mycolic acid. So this waxy mycolic acid during Zn staining is dissolved by heating, by applying heat. And examples of the AFBs we have is Mycobacterium tuberculosis, MTB. Then we have also Mycobacterium leprae and Mycobacterium usans. So these are some of the examples of the acid-fast bacilli. For them, they are demonstrated using Zn staining, whereas these ones are using gram stain, and we have seen how we can differentiate them. So thank you so much for following. Hope you have seen the differences majorly between gram-positive and gram-negative according to their cell envelope. which contains different layers. That is the capsule, the peptidoglycan layer, and the inner layer. That is for the gram-positive, together with the teichoic acid. Whereas gram-negative, they have the capsule, they have the outer layer made up of lipopolysaccharide, or you can call it endotoxin, which provides protection and typing for the gram-negative bacteria. And it offers virulent factors. That's why gram-negative bacteria, they are more virulent due to presence of lipopolysaccharide, which it is lacked in gram-positive. It has a periplasmic space and at the same time inner membrane. And we have said within the periplasmic space, we have lipoproteins which anchors the outer membrane to the peptidoglycan layer. It has the hydrolytic enzymes like the beta-lactamases, which destroys the beta-lactam ring of the penicillins and the cephalosporins. Thank you so much for learning with us. May the good Lord bless you.